Respected Dean Sir, I'm thankful to you for sparing your valuable time and joining this examination by voice. I welcome Dr. Subhash Sharma for accepting and for meeting us, and we are honored to have you here as an external examiner for the student, PhD student Dr. Ankit Jain. I'm also thankful for joining the voce Dr. Suprema, Dr. Aij, and Major by Dr. Joyce Jogi. So first of all, Dr. Ankit, you give your introduction and then start the presentation.
Thank you, ma'am. I'm Dr. Ankit Jain. As a student, I joined the PhD in the 2021 batch. I completed my PhD from this college. My UG was started from 2005 to 2010 from this Novo Veterinary College. Then I completed my PG from Girvasu, Ludhiana, and then I was posted in the field since 2013, and then I joined this college. My topic of the study during my thesis work was the study on the bacterial biodegradation of low density polyethylene. So my major advisor was Dr. Joyce Jogi ma'am. Under the guidance of Dr. Joyce Jogi ma'am and the advisory committee, Dr. Daljit Chawda ma'am, Dr. Supriya Shukla ma'am, and Dr. Ranjit sir, I completed my research on this topic. So, welcome to Dr. Subhash Sharma sir, external examiner, then respected Dean sir, respected major advisor and advisory committee. I am going to present my thesis seminar on the topic studies on the bacterial biodegradation of low density polyethylene. So plastic pollution is a major concern, and it not only affects the environment, it also affects the human beings and animals. The animals were died due to ingestion and choking with this plastic material. And so many researchers have tried to mitigate this pollution. Hence, there are several management practices like recycling, burning, and incineration, and dumping into the ground are the techniques. But even though these are not sufficient to mitigate this plastic pollution, so many researchers are tried to mitigate this plastic pollution by eco-friendly way and economical way. From different abiotic components, so many bacteria were also isolated to degrade the plastic material, and here we are trying to isolate the bacteria from the biotic component which are capable to degrade the plastic. So looking to that, during my research work, the study was conducted on the following objectives. The objectives was isolation and identification of low density polyethylene degrading bacteria to determine the biodegradability of low density polyethylene, comparison of degradation ability of different biodegrading bacteria. So to fulfill these all requirements, the work was done at the Department of Veterinary Microbiology, College of Veterinary Science and Animal Husbandry, Mahu. The first objective was isolation and identification of low density polyethylene degrading bacteria. To fulfill this objective, the LDP were selected, and according to the classification reported by Mariotti et al., and then further it was measured by micrometer caliper. 50 micron thick LDP were selected for further biodegradation study. Then these plastic were further treated with the benzene to remove any plasticizer and coloring agent. Then these LDP were treated, cut into the 1.5 into 1.5 centimeter square piece, then 1.5 centimeter diameter circle, then 2 centimeter diameter circle, and 1.5 centimeter triangles for different biodegradation experiments. These all pieces were treated as according to Lee and co-workers 1991 and Yaw and co-workers 2022. These LDP were treated with the UV rays at 365 km for 48 hours, and further they will be stored in a room temperature in desiccator. Then the LDP powder were also prepared according to Nanda and co-workers and Bhatia and co-workers, and again the UV exposure was given at 360 nanometer for 48 hours, and this powder were again stored for further biodegradation experiment. Then the samples were collected from veterinary clinical complex and instructional livestock farm complex, veterinary college Mahu. These samples were fecal sample of cattle, buffalo, goat, horse, poultry, pig and dog, and sewage water, dumping ground, soil, garden soil were also collected from different dumping area and polluted area in and around Mahu. So these are the list of the samples that were collected during from different latitude and magnitude, and they were also given the lead numbers. Total 10 samples were collected. During the whole degradation study, these media were used in the study, like the MSM broth with 0.2% LDP powder. It was used for the enrichment of bacteria. Then MSM agar with 4 LDP pills and 0.2% LDP powder. It was used for as a basal media for the isolation of bacteria. Then MSM agar with 0.2% polyethylene glycol. It was used for clear zone analysis. Then MSM agar and MSM broth were containing LDP film. They were used for further biodegradation study. Then nutrient agar and urea verteni glycerol broth were used for the maintenance of these isolates. And further, salmon citrate agar, glucose phosphate broth and peptone water, they were used for biochemical study. For the isolation of LDP degrading bacteria, first the enrichment broth were prepared by inoculating 1 gram of the sample and containing 0.2% LDP powder into that. It was incubated at 120 RPM for 30 days in the shaker incubator at 37 degrees centigrade. And further, after 30 days, presence of bacterial growth were stained by gram staining. Presence of bacterial growth were measured by the gram staining. Then further, this enriched broth were inoculated onto the MSM agar plate containing LDP film and 0.2% LDP powder, and these sample were again incubated for 60 days at 37 degrees centigrade, and the presence of bacteria were closely observed and gram staining were performed. Then the identification done by microscopically and biochemically is followed by Bell and Peltham, Malik and colleagues and co-workers. Comes to the screening. These isolates were screened by the clear zone analysis according to Rana and Rana. In that MSM agar were containing 0.2% polyethylene glycol were used. They were incubated for 14 days, and then after 14 days, the staining was done by 0.1% commercial brilliant blue solution, and this staining done by this staining solution, and then those isolates which are showing clear zone around the colonies were selected further for the study. Then the identification was done by the BD-Phonics M50 automated identification and susceptibility test system, and by the molecular characterization using 16S rRNA gene by universal forward and reverse primer by sending these isolates to Barcode Bioscience, Bangalore, Karnataka, India. Then further, the sequencer were submitted to the NCBI by querying the sequence by bioedit software, and then the accession number were obtained. Further, they will be analyzed for phylogenetically by using Mega 11 software using neighbor joining method, considering 1000 bootstrap values. Now comes to the result first. After 30 days of the incubation, this is the plate showing presence of gram positive, gram negative and pleomorphic bacteria into the enrichment broth. Then total 28 isolates were isolated from the MSM agar plate. Out of them, 12 isolates were gram positive and 16 isolates were reported as gram negative, and these isolates were given as isolate number. Then these 28 isolates were further characterized biochemically. So the presence of bubbles into the hydrogen peroxide showed positive for catalyst test. Change in the color from purple, white to purple showed presence of positive for oxidase test. Then red color ring on the surface of broth showed positive for indole test. Then change in the color from yellow to red showed positive for methyl test. Then light red ring over the surface of rod showed positive for Vegas posture, and change in the color from yellow to green to blue showed positive for citrate utilization. Then total 5 isolates were selected based on clear zone analysis. These isolates showed triple positive clear zone, and they will be further selected for biodegradation study. So out of 28 isolates, 5 isolates showed triple positive, and they will be selected. These isolates were M1, M4, P9, S1 and S2, and they will be characterized by Bidifonics M50 automated system and by sequencing. And they were Staphylococcus clausi, Cucuria rosea, Pseudomonas aeruginosa, Cellulosis microbiome cellulans, and Leuconostoc mesenteritis. And these are the accession number were obtained from the NCBI. And these plates are showing phylogenetic analysis of Staphylococcus clausi M1. This is the phylogenetic analysis of Cucuria rosea M4. This is for Pseudomonas aeruginosa. This is for Cellulosis microbiome cellulans S1. And this is the phylogenetic analysis of Leuconostoc mesenteritis S2. So finally, after the phylogenetic analysis, they were showing the homology percent similarity up to 90 to 99% with their subject sequences that were already present to the NCBI. So these are the colonies that were present on the LDP film, and this is a colony of Staphylococcus clausi M1. This is a this is a inoculated nutrient agar plate showing Staphylococcus colonies, and this is the gram staining showed gram positive foci. This is the colony of Cucuria rosea on the LDP film. This is the Cucuria rosea nutrient agar and gram positive cocobacilli of the Cucuria rosea. This is the colony of Pseudomonas aeruginosa on the LDP film, and this is the nutrient agar plate showing Pseudomonas aeruginosa colonies, and these are the gram 80 rods in the gram staining. Then these are the colonies of Cellulosis microbiome cellulans on the LDP film, and these are the colonies on the nutrient agar and gram positive rods in the gram staining. Then these are the pinpoint colonies of Leuconostoc mesenteritis on the LDP film, and these are the colonies on the nutrient agar, and these are the gram positive foci of Leuconostoc mesenteritis in gram staining. These all isolates were showing pigmentation ranging from Staphylococcus clausi, yellow, pink, then green was reported by Pseudomonas, then again pink and the white were reported by Leuconostoc mesenteritis. Comes to the conclusion that Staphylococcus clausi M1, Cucuria rosea M4, Pseudomonas aeruginosa P9, Cellulosis microbiome cellulans S1 and Leuconostoc mesenteritis S2 isolated from cattle feces, sewage water, poultry droppings, cattle feces and buffalo feces respectively were all effective in degrading low density polyethylene. Comes to the second objective, to determine the biodegradability of low density polyethylene. The experimental setups were conducted. Out of them, total 24 petri plates were taken by arranging 4 LDP films. Then further it were inoculated with 5 isolates and 1 control. They will be grouped into they will be incubated for 15 days, 30 days, 45 days and 60 days of the incubation period. And the inoculum was prepared according to 0.5 mg per length standard. The same degradation study were performed into the broth, and they were again incubated for 60 days containing 0.5 mg per length standard inoculum for 60 days. At different incubation period ranging from 15, 30, 45 and 60 days of the incubation. Then after completion of the incubation, these LDP films were collected at 15, 30, 45 and 60 days of incubation. These LDP films were analyzed for gravimetrically, then functional group analysis was also done, and scanning electron microscopy were also done. For gravimetric analysis, the collection of LDP were done according to Harshvardhan and Jha, and the gravimetric analysis was conducted by this following formula. Then further the gravity analysis was conducted by using ANOVA as described by Isner and Cochran. Comes to the result that LDP 1, 2 and 3 that were collected from control group from the MSM agar plate, showing that initial weight, final weight, weight loss and weight loss percentage at the different incubation period is reported in this table 9, and the same is collected from the MSM broth, the control LDPE that is showing in this table. Then LDP that were collected from the after the degradation by Staphylococcus clausi M1 is reported into this table at different incubation period, and LDP and the same is collected from the MSM broth. This is collected from the MSM agar done by the Cocoria rosea, and this is from the MSM broth. The same that is the table showing collection of this LDP film after the different incubation period degraded by Pseudomonas aeruginosa from the MSM agar, and this is from the MSM broth. This is from the MSM agar degradation done by Cellulose microbiome cellulans, and this is from the MSM broth. This is the degradation of LDP were reported by Leuconostoc mesenteritis S2 into the MSM agar, and the same were reported by the MSM broth. These are the initial weight, final weight and weight loss is graphically presented here. Showed that the Pseudomonas aeruginosa and Cellulose microbiome cellulans showed highest degradation among all isolates at the 15th day of the incubation period. Then again the Pseudomonas aeruginosa showed highest degradation at 30th day of incubation period. Then Pseudomonas showed highest at 45th, and again on 60th day it was reported highest degradation among all isolates. Then into the broth, again the Pseudomonas aeruginosa and Cellulose microbiome reported similar degradation at 15th day of the incubation period. Then the Pseudomonas reported higher than Cellulose microbiome cellulans and reported at 30th day of the incubation period. Then at 45th again it was higher, and at the 60th end of the degradation experiment it was highest reported by Pseudomonas aeruginosa. Then the ANOVA table showed that among different days all isolates were reported significant weight loss except Cocoria rosea M4 and Cellulose microbiome cellulans S1. The same is representing in this table that the significant weight loss reported by Staphylococcus clausi into the MSM agar at different incubation period, and it was highest on the 60th day in the MSM agar. Then it was reported that into the MSM broth among different days all isolates were reported significant weight loss, and the same is reported into the graph that Staphylococcus clausi M1 reported significant weight loss at different incubation period into the MSM broth, and it was highest on the 60th day of the incubation period. The Cocoria rosea showed non-significant weight loss, and it was highest at the 45th day of the incubation period into the MSM agar. It was significant weight loss reported by Cocoria rosea into the M broth, and it was highest at 60th day of the incubation period. Then Pseudomonas aeruginosa showed significant weight loss into the MSM agar, and it was highest on the 60th day of the incubation period. Then Pseudomonas aeruginosa canine showed significant weight loss into the MSM broth, and it was highest at the 60th day of the incubation period. Then Cellulose microbiome cellulans reported non-significant weight loss into the MSM agar, and it was highest at the 60th day of the incubation period. Then Cellulose microbiome cellulans reported significant weight loss into the broth, and it was highest at 60th day of the incubation period. Then Leuconostoc mesenteritis showed significant weight loss into the MSM agar, and it was highest at the 60th day of the incubation period. Then Leuconostoc mesenteritis showed significant weight loss into the MSM broth, and it was highest at 60th day of the incubation period. Now comes to the conclusion that LDP biodegradation increased significantly over time. Now to fulfill the objective third, that was the comparison of degradation ability of different biodegrading bacteria, again the ANOVA was conducted according to Isner and Cochran, and further it was analyzed by FTIR and SEM for the functional group analysis and surface analysis respectively. For the functional group analysis, the collection of LDP film were done according to Osmier and co-workers, and it was analyzed by as per reported by Socrates by seeing the change in the absorption peak of the functional group. These collected LDP film were sent to Sophisticated Analytical Instrumentation Facility IIT Mumbai. They will be analyzed on three broad bases. First, UV untreated LDP film and UV treated LDP film were compared for functional group analysis. Then UV treated control LDP film before incubation and after incubation into the MSM agar and MSM broth were compared. And third, UV treated LDP film before incubation and after degradation by the isolates were compared at the end of 60th day of the incubation. Then carbonyl index were also calculated as reported by this formula that was reported by Albertson and co-workers. And either increase or decrease in the carbonyl index were reported as it was indicated as LDPE degradation. Then the collection of LDPE for surface analysis was done according to Harshvardhan and Jha, and this collected LDPE film were sent to Sophisticated Instrumentation Center IIT Indore and SIF IIT Mumbai for presence of any crack groove, then any hole formation on the LDPE film and presence of any bacteria on the surface. Comes to the result third, comparison of biodegradability of isolates in MSM agar at each incubation period. So it was found that among different isolates at different incubation period, it was found significant except at 15th day of the incubation period into the MSM agar. That the degradation was non-significant at the 15th day of the incubation period into the MSM agar, but and the same is showing here that it was highest by the cellulose microbiome cellulence S1 at 15th day. But it was found significant after 30 days of the incubation period, and it was highest by the Pseudomonas aeruginosa. Then at the 45th day, it was again significant, and it was highest reported by Pseudomonas aeruginosa. Then at 60th day of the incubation period, it was significant, and it was highest reported by Pseudomonas aeruginosa among all isolates. So these are the comparison of different biodegradation done by different isolates at different incubation period, and it was showed that Pseudomonas showed highest degradation at 60th day of the incubation period into the MSM agar. Now comes to the degradation comparison into the MSM broth at each incubation period. So it was reported that among different isolates at different incubation period, it was significant degradation. And this significant degradation was reported at 15th day of the incubation period among different isolates, and it was highest by Pseudomonas aeruginosa at 15th day. It was significant at 30th day and highest reported by the Pseudomonas aeruginosa at 30th day of the incubation period. Then the degradation was significant at 45th day, and again it was highest at, done by the Pseudomonas aeruginosa into the MSM broth, and again significant degradation was reported by the different isolate into the MSM broth at 60th day of the incubation, and it was highest reported by the Pseudomonas aeruginosa. These are the comparison of different isolates showing degradation at different incubation period, and it was showed that highest degradation was reported into the MSM broth that was done by the Pseudomonas aeruginosa. Now coming to the comparison of biodegradability of isolates in between media. So it was reported that non-significant weight loss was seen in the control LDP film at 15, 30, 45 and 60 days of the incubation period. But after that, if we compared the different isolate performance of degradation in between media, it was reported non-significant at 15th day, then again non-significant at 30th day, then at 45th day it was significant only reported by Liponestock mesenteroides, and at 60th day it was reported significant by Bokuria rosea M4. So among all different incubation periods in different media, it was seen, it was shown that the non-significant weight loss was reported by the isolates. So these are the tables showing that non-significant degradation was reported by Staphylococcus chlorsi at 15, 45th, 30th, 45th day of the incubation period and 60th day of the incubation period. And it was highest in the MSM Roth as compared to MSM Agar at 60th day of the incubation period. Then from the Cucuria rosea, it was non-significant at 15, 30, then 45, and significant was at the 60th day of the incubation period, and it was highest reported into the MSM Roth at 60th day of the incubation period. Then from the Pseudomonas aeruginosa, it was again non-significant at 15, 30, then 45th and 60 days of the incubation period, and it was highest at 60th day of the incubation period into the MSM Roth as compared to MSM Agar. Then non-significant degradation reported by cellulose maculum cellulence S1 at 15th, 30th, 45th and 60 days of the incubation period, and it was highest at the 60th day into the MSM Roth as compared to MSM Agar. Then again non-significant degradation were reported by leuconostoc mesenteroides into the MSM Agar and MSM Roth, and at 15th, 30th, then it was significant at 45th and again non-significant at 60th day of the incubation period, and it was highest at 60th day into the MSM Roth. So it was a comparison reported in the graph showing that into the MSM Agar and into the MSM Roth, the highest degradation was reported by Pseudomonas aeruginosa into the MSM Roth at 60th day of the incubation period. Then the FTIR was analyzed in three major bases. In between UV untreated and UV treated LDP film, it was reported that the three characteristic peaks were reported by UV untreated LDP film before incubation and before degradation, and these three major peaks were reported at different wavelengths, and this was of CS2, then CH and CS2 bone absorption peak. The same is representing into the graph that these are the three major characteristic peaks were reported of the LDP film before starting of experiment. Then after experimentation, when the UV treatment was given to the control LDP film, there was change in the absorption of intensity is reported, and the same is showing into the graph that the red one indicated the UV treated LDP film before incubation as compared to blue line showing that UV untreated LDP film before incubation. But after incubation, it was reported that there were no any absorption peak change before incubation and after incubation into the MSM Agar and MSM Roth in the control LDP film. But when they were treated with Staphylococcus clausi M1, there were change in the intensity of absorption peak were reported into the MSM Agar, and the same is presenting into the graph showing that the red line is showing the LDP that were treated with the Staphylococcus clausi and compared with the blue line of the respective control. The change in the intensity of absorption peak were reported by Staphylococcus clausi into the MSM Roth, and the same is presenting into the graph. Then the change in the intensity of absorption peak were reported by Cucuria rosea in the MSM Agar, and the same is presenting into the graph. Then change in the intensity of absorption peak of Cucuria rosea that were reported by into the MSM Roth, and the same is presenting into the graph. Then Pseudomonas aeruginosa showed also change in the intensity of absorption peak of LDP film into the MSM Agar at the end of 60th day of the incubation period, and it was characteristically seen that the major degradation was reported by the Pseudomonas aeruginosa also in the FTIR. Then into the MSM Roth, again Pseudomonas aeruginosa showed change in the absorption peaks of the LDP film, and characteristic change is also reported in the graph. Then in the MSM Agar, cellulose maculum cellulence showed a change in the absorption peak, and it was also reported into the graph. Then change in the absorption peak were reported into the MSM Roth by cellulose maculum cellulence, and the same is shown in the graph. Then change in the intensity peak were reported by leuconostoc mesenteroides into the MSM Agar, and the same is reporting into the graph. And into the MSM Roth, again the same intensity were change in the intensity were also reported, and the same is presented into the graph. Then after the FAR analysis, the carbonyl index were calculated according to the formula as reported by Albertson et al. And it was it was shown that before UV treatment, the control LDP showed 4.82 carbonyl index, and it was further increased after the treatment with the UV, and it was reported that 12.67 means there were increase in the carbonyl index before degradation. When this increased carbonyl index that were treated with the UV LDP film were subjected for degradation with the following isolate, it was reported that again the decrease in the carbonyl index was reported, and it was highest degradation by highest decrease in the carbonyl index were reported by the Pseudomonas aeruginosa into the MSM agar, and it was 5.21. Then into the MSM broth, the 4.77 of carbonyl index were reported by the Pseudomonas aeruginosa. Then the percent decrease of carbonyl index were calculated by this formula, and it was reported that Pseudomonas aeruginosa showed UV treated showed highest decrease in the carbonyl index into the MSM broth, and it was 62.32%, and in the MSM agar it was 58.82%. The same is represented into the graph that it was highest reported by the Pseudomonas aeruginosa in the broth. Then surface analysis were conducted, and after the end of 60th day of the incubation period, and it was showed that the control have smooth surface at initial experimentation, initial before experimentation, and after experimentation at the end of 60th day of the incubation period, the control showed smooth surface that were collected from the MSM agar, and that were collected from the MSM broth. These were also showed smooth surface. But the degradation were reported by the Staphylococcus glossy M1 into the MSM agar, and there were surface erosion were found on the surface of LDP film, and there were hole formation and aberrations were reported by Staphylococcus glossy in the MSM broth. These are the presence of Staphylococcus glossy that were collected from MSM agar, and they will be collected from the MSM broth. Then the surface erosion reported by the Cocoria rosea in the form of hole formation, pit formation, and crack formation was there from the MSM agar, and this is the plate showing from the MSM broth. These are the presence of Cocoria rosea on the surface of LDP film after end of the 60th day of the incubation period, and this is from the MSM agar, the presence of Cocoria rosea that were showing degradation of LDP film also reported in this plate. Then surface erosion were reported by Pseudomonas aeruginosa in the form of surface aberration, then hole formation, pit formation, and aberration was there. And this is the picture of that were collected from the broth showing the hole formation on the surface of LDP film. These are the presence of Pseudomonas aeruginosa, and clearly it was seen that the presence of cracks around the colonies was there. These are the LDP film were collected from broth, and it was reported presence of cracks with presence of bacteria on the LDP surface. These are the degradation reported by cellulose microbiome cellulence that were collected from MSM agar, and this is where from MSM broth. These are the presence of cellulose microbiome cellulence on the MSM on the LDP film surface that were collected from the MSM agar, and this were from the MSM broth. These are the surface aberration were reported by the Leuconostoc mesenteritis from the MSM agar, and this were from the MSM broth showing that hole formation and the surface erosions was there. These are the presence of bacteria on the surface of LDP film that were collected from the MSM agar, and that were collected from the MSM broth. Now comes to the conclusion that the biodegradability of all isolate was reported higher in the MSM broth than MSM agar, and Pseudomonas aeruginosa P9 showed the highest LDP degradation in both media. The increase in the carbonyl index by UV rays followed by a decrease was due to bacterial biodegradation. Then comes to the overall conclusion that Staphylococcus clausi M1, Cochlear rosea M4, Pseudomonas aeruginosa P9, Cellulose microbiome cellulence S1 and Leuconostoc mesenteritis S2 isolated from cattle feces, sewage water, poultry droppings, cattle feces and buffalo feces respectively were all effective in the degrading low density polyethylene. Then LDP biodegradation increased significantly over time, and the third conclusion was the biodegradability of all isolate was reported higher in MSM broth than MSM agar, and the Pseudomonas aeruginosa P9 showed highest LDP degradation in both media. The increase in the carbonyl index by UV rays followed by a decrease was due to bacterial biodegradation. Then suggestion for further work is a study on other biotic component for isolation of LDP degrading bacteria can be done. Then a study on biodegradation of other plastic like polyethylene terephthalate, high density polyethylene, polyvinyl chloride, then polypropylene, polystyrene can also be done. Then a study on bioenzyme degradation of low density polyethylene and other plastic can be done. Then a study on the plastic degradation using combined microbial consortia can also be done. Exploration of gene associated with the plastic degrading enzyme and characterization of biodegradation associated with the plastic degrading can be done, and degradation kinetic using other environmental factors like temperature, pH, salt concentration, incubation period, and growth media can also be studied. Thank you.
Okay, thank you, Dr. Ankit.
Thank you.
Questions are invited from the advisory and the external examiner.
Yes, so thank you for a nice presentation.
Thank you, sir.
So I just want to make few remarks. The presentation was very nice and you have presented the results very nicely.
Thank you, sir.
And you are very thorough. And you have done almost everything that needs to be done with this kind of work. Because I am also not aware of this kind of work. This is new to me because hardly anyone in microbiology works on this kind of problem. So first of my question is like, why did you choose this as your PhD problem as a student of bacterial microbiology? So that's good. So like when you were thinking of mitigating this problem, we started your PhD problem. So how do you think that you're going to mitigate? Like once you do this kind of work, how you
Use the methane from the gobar gas. So the bioreactor will also provide us some consumable item like water and carbon dioxide that will be produced during this bioreactor. So it may be possible in the future that the bioreactor will work for mitigating this plastic material.
That's okay. I have some general questions. So, because you saw that your biodegradation happened best in the broth.
Yes, sir.
So, what could be the reason?
Sir, there may be the chances of nutrient availability will be more in the broth. First. Second, the enzyme solubility will be the higher into the broth. Third, the surface, both surface of the LDP film is available to the bacteria for the degradation. That's why it was higher in the broth as compared to the agar. And the fourth one is that the presence of biofilm formation is more higher in the MSM broth because the both surface will be available to the LDP film to the bacteria.
And did you also check the biofilm production in the bacteria? You did not check?
No.
should not do that kind of experiment.
No, sir.
Okay, so another question is, what is the best way to determine which of these bacteria, quickest way and best way now to say that if we want to screen the bacteria for their plastic biodegradability, what method would you suggest? One or two methods? What would be the priority method first to do, and then maybe second? So if you want to suggest two methods.
Sir, first we have to go for enzymatic analysis of the different bacteria. So
Did you do that enzymatic analysis? Because you didn't do so.
no, sir.
Based on your methods, I'm saying. If the enzymatic method was the best, then why didn't you do that?
No, sir, because of the lack of time, and it was time.
You did all the good essays. So among these essays, which one will be the best?
Sir, FTIR will be the best.
FTIR will be the best, or the one which you did maybe agar zone, clearance zone analysis?
Yes, sir. Clear zone analysis is the primary method to screen the isolates.
So that's what I'm asking. If you want to screen the isolates
On the basis of clear zone analysis.
clear zone analysis. Okay. And second?
And second, sir, it was the FTIR. It was treatment.
FTIR is more advanced method. I don't know what you have done something.
Sir, it was cheap actually.
Cheap.
FTIR is also cheap.
But it's not available everywhere. You have to send your samples to IIT for FTIR.
Yes,
State.
yes.
So this facility is not available everywhere. So you went yourself with your samples?
Yes, sir.
Posted them.
No, sir, I went my samples to IIT Indore and I posted to Mumbai.
Right. Okay, the treated and untreated all.
Yes, sir.
Just in a normal courier.
Yes, sir.
Okay. So you were suggesting you could do some enzymatic assays. What are those enzymatic assays, or some kind of colorimetric assays you could do?
Sir, it cannot be. I don't know, sir.
So what these enzymes which are responsible for biodegradation? Can you name these enzymes?
Yes, sir. Like esterase, petanes, cutinase, and
न्यूट्रिशन को दे दिया फोन करके
sir, esterase and petanes is mainly responsible for.
Lactase.
Yes, sir.
Lactase and aspartame.
Yes, sir. Because polyethylene terphthalate was also there.
So how do you screen for these enzymes? Can you suggest some assay?
Sir, maybe colorimetrically. I have no idea, sir.
And you were also talking about you can look for expression of genes.
Yes, sir.
So what are those genes?
Sir, the genes which are responsible for producing such type of enzymes which are responsible to degrade the LDP.
Another way of looking at these isolate could be to screen them for these genes in the first place. If they are present in these bacteria or not. So that would be one quick assay by PCR you can also screen.
Yes, sir.
And then obviously phenotypic assay you are suggesting that is best way to analyze functional characterization.
Yes, sir.
Genome levels you can do PCR to screen. Okay, and if you want to increase their plastic biodegradability potential, what could be the alternative
Sir, we can-
interventions that can be done?
Sir, we can enhance the first UV exposure. In my experiment, it was given only for 48.
UV exposure is, if you want to make a bacteria, for example, you found Pseudomonas aeruginosa. It was aeruginosa?
Yes, sir.
So because the 20% biodegradability close to that you saw,
Yes, sir.
is this a good candidate for further industrial application to be taken for bioreactor kind of scalability?
Yes, sir. Definitely. Out of these isolates, the Pseudomonas aeruginosa performance of Pseudomonas aeruginosa was higher.
But
So
is it industrially viable
No.
solution?
no, sir. We have to express the particular gene for production of particular enzyme, and that we have to insert it into a cloning vector for the production and expression of the enzyme. It may be useful for the industrial area.
So what should be the normal threshold for a bacterium to be a industrially acceptable for plastic biodegradability? How much
Sir,
should be able to do?
Sir, it should be non-infective, non-hazardous to the not only to the human being and animals but also to the environment.
So my question is like how much potential, like you said it's 20%,
Yes, sir.
40%, 80%, how much time, because it took 60 days.
Yes, sir.
This is not,
No, sir. We have to reduce the time by giving the exposure of UV rays to 1 to 2 months. So it will degrade quickly within. I saw the literature. Within 15 days, it may be degraded up to 50 to 60%.
and do you think it is going to be a successful in the future in comparison to now what they do is chemical treatment for degrading the plastic and all industry uses?
Sir, we have to go to both techniques because chemically we will find out different toxic gases are also producing, and this can also be mitigated by the bacterial digestion. Otherwise, these toxic gases will create the pollution in the environment. So we have to apply both all the techniques to reduce the pollution. We have to go through all the steps like chemical and the biodegradation also.
So how could you increase the interaction of your bacterium which is plastic biodegrading to the plastic, because plastic is hydrophobic?
Yes, sir.
So one of the thing is that these bacteria are not able to attach to the plastic. So can you do some kind of interventions?
Yes, sir. Like digestion with different chemicals like the SDS, P90. These chemicals were enhance the.
But it will become expensive to the plastic. The whole process will become expensive and then you are going to use chemicals, so the whole process will be difficult. Is there any way like genetic manipulation or anything that can be done? Because also these bacteria are slow growing and we have only looked at the aerobic microflora. Many of the plastic biodegrading maybe are anaerobic.
Yes, sir.
So that aspect we have missed in here, isn't it?
Yes, sir. Yes, sir.
And obviously in the gut, the environment in the gut is entirely different in the inside the cow's stomach. So we are not able to replicate those conditions outside.
Yes, sir.
Obviously there may be a very good consortium of bacteria inside, but we are not able to find them out in the lab.
Yes,
So if we want to genetically manipulate Pseudomonas aeruginosa, what different genetic manipulations can be done? Like if you can say point wise.
Sir, we have to manipulate to become it act on hydrophobic material also.
So for that, what would be the which pathway will be targeted? Any idea? Okay. What next?
No, sir, no idea.
So you can read these aspects also. And one way is also to take these genes, which are responsible for biodegradation, is to clone them and put it into bacteria and put it into E. coli or something else.
Yes, sir.
Because E. coli is easy to grow, we understand the biology and everything.
Yes, sir.
And we can put in very strong promoters that can enhance the production of these enzymes.
Yes, sir.
And other metabolic pathways or operations can be done.
Yes, sir.
So, because that is how companies are going to sell their product. They will say that we have this patented formulation consortium because they have genetically manipulated it. Then they will sell it as a environmental friendly measure to degrade plastics. This is a good area for it. It's only people who can brand and you can produce it, they can make a lot of money out of it. So I have only these questions. Thank you very much.
Thank you very much, sir.
Dr. Ankit, any idea of the cocktail of the bacteria and
Yes, ma'am. Actually, the second was the performance of cellulose microbiome cellulence was higher as compared to the other isolates. So we have to prepare a cocktail of these bacteria. We have to again screen such type of isolates like cocktail of again Pseudomonas aeruginosa should be done by different samples, and we can apply it for the future experiments.
To my knowledge, there were so many bacteria isolated, but we proceeded only for.
Yes, ma'am. It was they will be isolated from abiotic components, and this was the first experiment conducted by using biotic component for the isolation.
Your statistically significant findings are the same as what you did in the microbiome. So that's some places I've seen. Mostly you have this the aeruginosa, you have said it's the best.
Yes, ma'am.
And but at some places your values are not they are not similar for what you are reporting individually. आपको कहीं ऐसा कहीं कुछ मिला है जैसे वेरिएशंस?
मैम कहीं कहीं मुझे ऐसा लगा है कि कहीं किसी बैक्टीरिया का परफॉर्मेंस ब्रॉथ में ज्यादा अच्छा था, किसी बैक्टीरिया का परफॉर्मेंस जो है अगार में ज्यादा अच्छा था. जैसे कुकुरिया रोसिया और सेलुलोसिस माइक्रोबियम सेलुलेंस का बीच में कहीं परफॉर्मेंस जो है ब्रॉथ में अच्छा रहा तो उसने सिग्निफिकेंट डिग्रेडेशन दिया है. तो मैम अभी इसमें बहुत सारी प्रोबेबिलिटी है कि हमें बहुत सारी डिफरेंट सिचुएशंस को स्टैंडर्डाइज करके और उसके बाद में हमें देखना होगा कि किस बैक्टीरिया का परफॉर्मेंस किस कंडीशन में ज्यादा अच्छा हो सकता है. ऐसा जरूरी नहीं है कि इस समय हमने जो यस मैम यस मैम वी हैव टू पुट ओनली yes ma'am. So we cannot able to only decide to degrade by the Pseudomonas aeruginosa. We have to prepare the cocktail for different environmental situations for the experimentation.
Okay, but it was a good topic from one health
Yes, ma'am.
point of view.